Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 153 for the podcast. Humacyte is a company worth watching. They are pioneering a platform that enables the investigation, development, and manufacture of bioengineered human tissues and organs that are designed to be universally implantable, off-the-shelf, and regenerative, with the goal of treating a wide variety of diseases, injuries, and chronic conditions. This week, I had the chance to speak to Humacyte founder, president, and chief executive Laura Niklason about the company she's built, the science she's driving forward, and the real-world applications for Humacyte's lab-grown blood vessels in some of the most devastating and dangerous conflicts. I hope you enjoy our discussion. Laura, welcome to Beyond Biotech.
Dylan, it's great to be here. Thank you very much.
I just wanted to start by getting you to introduce yourself to our audience a little bit and tell me how you actually moved from, growing up in Illinois right through teaching, research, to eventually founding Humacyte. Talk to me about that journey.
Well, I guess it's a long answer to that, but when I was a little girl, I always wanted to be a physician and I wanted to invent things. Those were two things that I very much wanted to do, and I would say that stuck with me all the way through adulthood. In my medical training, I trained as an anesthesiologist and an intensive care unit doctor because I was very interested in caring for the very ill. And I was also very interested in trying to figure out how we can support patients better who have tissues and organs that are failing. And that really led me into the whole field of tissue engineering in the mid-90s, which is what we now call regenerative medicine. And because vascular disease kills so many people, it kills more people than any other disease, more than cancer. I became very interested in trying to figure out if we could grow replacement blood vessels for humans. And so that's what I've spent the last 30 years trying to do.
Like you say, you've been working in this field for decades. Was there something particular that drew you into the vascular world?
Well, I guess I would say that, again, blood vessels are so central and important, obviously, to human health. And I think we all know that. But there is one particular case. I was taking care of a patient when I was a resident in Boston at Mass General. And this was a patient who was having a heart bypass. And to do a heart bypass, to replace clogged arteries on the heart, we typically take veins out of the leg to do those replacements. And so I can remember I was standing at the head of the bed and taking care of this patient, and the patient, the chest was open, and the surgeons went to the right leg and opened up the right leg and looked at the vein and said, "That's no good," and closed up the incision. They went to the left leg, opened up the left leg from top to bottom, looked at the vein, said, "That's no good," and closed it up. They then went to the left arm, opened up the arm, thinking they would take an artery out of the arm, decided that wasn't good, closed that incision, and then they made a fourth incision, and they basically extended the incision from the chest into the abdomen and took a vessel off the stomach to put it onto the heart. And watching that whole process, it really seemed barbaric. It seemed like we were doing so much damage to the patient in efforts to help the patient. And I just became very interested in trying to answer the question, well, can we, instead of injuring the patient to get a vein, can we grow a new artery for someone who needs one?
It's incredible, really. I mean, this is one of the top hospitals. I assume some of the top surgeons as well. And we're still digging around just trying to find a vein that we can use.
Well, and not only that, but, heart bypass surgery started in the 1960s. And in the 1960s, we were digging around in the leg to take out vein to do the bypass. It's now 2025, and that's exactly what we're doing 65 years later. So certainly surgical techniques have gotten better. But, the whole area of finding a blood vessel replacement, that's been a really hard problem.
And this is where Humacyte comes in. You started with, a $150,000 loan in 2006. Now you're a public company. How did you actually scale? And I guess to go back to the start, what's the mission of Humacyte?
Well, the mission of Humacyte is really to be able to provide to patients and surgeons uh, something I like to call spare parts for people. These are replacement tissues, initially blood vessels, but we hope to grow other types of tissues over time. Blood vessels that can be literally taken off the shelf and used in any patient at the time that the patient and surgeon need them. So that's really our mission. As far as the start, I had been working in this field for about 10 or 12 years. When in 2005, after I had moved to Duke University, I decided to spin out Humacyte because I felt like we had a line of sight. We hadn't solved all the scientific problems, but I felt like we had a line of sight to being able to put together a recipe for growing new human blood vessels. And so I started with just a couple of my students and a technician, and there was four of us, and we got a loan. But we also, frankly, took money from friends and family for years, and we wrote grants to the NIH and to other departments in the federal government, to the Defense Department, and we just bootstrapped it up and cobbled it together.
Is this a real niche player, something very unique, or are there others working in this regenerative space as well?
There are certainly other companies that are working in regenerative medicine and even in regenerative, blood vessels. I'd like to say, and I think it's true, that we're about 10 years ahead of anybody else who's trying to do this. Being a public company that just became public a couple years ago, sometimes I think of us that we were sort of flying under the radar for a number of years. And when we became public, then it became clear to a larger number of people what we were doing. And we're pretty far ahead of the pack, I've got to say.
And you're mainly working in the United States, or you've got a global reach as well?
So our clinical trials have been done in the United States, multiple countries in Europe, and we've also treated war wounded people in Ukraine. We started doing that a couple years ago. Currently, we are approved our engineered blood vessel, which we call the ATEV, which stands for acellular tissue engineered vessel. The ATEV is approved for use in patients with traumatic injury in the U.S. only.
Okay. You're also partnered, if I read this right, with Fresenius Medical Care, and they work in dialysis. Has that helped you to scale as well? Have you been working closely with them?
Well, Fresenius became interested in Humacyte and our engineered blood vessels really as a potential new conduit for patients who have kidney failure who are on dialysis. Because when you're on dialysis, what you need is a connection between an artery and a vein that nurses can access with needles and perform dialysis 3 times a week. We've been testing our blood vessel as an access point for hemodialysis for more than a decade. And Fresenius, when they've seen some of our data, has been very interested in that. So yes, they're a wonderful partner for us. And they've certainly helped us understand the landscape of dialysis patients and which patients are in the greatest need for having better access.
I guess it says something about the importance of vascular health that you're working in dialysis, you're working in traumatic injuries, you're in humanitarian aid over there in Ukraine as well. I mean, is there a limit to what you can work on?
Well, I'd like to say no, that there's no limits. But I will say that in addition to the trauma clinical trials and dialysis clinical trials, we've also conducted phase 2 trials in peripheral artery disease, which is where the arteries in the leg are blocked, and that can cause pain and can lead to amputation. We've done some very exciting work there showing that we can help many patients avoid amputation and keep their limbs. We've also been working in animal studies with a smaller version of our blood vessel that's actually the right size for a human heart. And it kind of takes us back to that case that I told you about 30 years ago, where we're now making blood vessels that we hope to test in heart bypass for patients sometime next year.
Maybe you can walk me through a little bit of the science. It sounds incredible, the sort of applications you have. But when it comes to the science, how do you create and grow these tissues?
Well, it really all starts with vascular cells. So, if there's anything that I've learned in the last 30 years, it's that human cells are a lot smarter than we are. They're very intelligent about the programs that they can execute and the way that they can make a new tissue, even in the laboratory or in our manufacturing system. But the key is to have the right cells and also to give them the right cues, the right set of stimuli so that they execute their program and go from being individual cells to being a whole blood vessel. So we've worked for decades to identify the right cells, the right vascular cells, and we actually have a bank now of vascular cells that can support our production for the next 30 or 40 years. So we've sort of got that piece nailed down, I think. And then over the last 20 years, we've really optimizing the conditions that we have to show these cells so that they become a human artery. So we have a degradable scaffold that the cells attach to, and that defines the size and the shape of the artery that we grow. We also, while the cells are growing in our bioreactors, we stretch them. We stretch them in a cyclic way, in a way that mimics your heartbeat, because every time your heart beats, your arteries stretch a little. And as it turns out, we learned almost 30 years ago that if we don't stretch these cells properly, they don't know that they're supposed to form an artery. It's an important cue. And so we also provide other growth factors and nutrients, and this is really the secret sauce that helps us guide these cells to form a new tissue.
How about the integration with the body, though, and actually becoming functional once it's implanted or surgically implanted? You know, it's implanted into a living person, living blood vessels, the rest of an entire system. You know, how does that work, the integration?
Well, the integration is another key piece of this technology. And going along with the integration, it's the lack of rejection. So, as I mentioned, we have a cell bank that we use to make these arteries. But what we also developed, in addition to figuring out how to coax the cells to make a new artery, after they make the artery, we also spent several years figuring out how to wash the cells back out of that artery, which sounds a little counterintuitive. But really, after we do that, the artery that's left behind is really just proteins. It's proteins that those vascular cells made, like collagen. And what we found is that if we do a good job of washing the cells out of the engineered artery, then we found two things. One, we can implant it into anybody, and we haven't had a rejection episode yet. We've treated more than 600 patients over more than a dozen years, and we've never seen an episode of rejection, which is exciting. But in addition, because this blood vessel, after it's implanted, has no cells in it, my vision is that it sort of looks like an empty apartment building to the cells in the body. And what we've seen over time is that cells from the patient migrate into that empty apartment building, that empty blood vessel, and turn it into a living artery over time, which is very exciting.
One of the big differences with your approach is that it's actually real human tissue, real human cells, because there have been synthetic ones before. What's the big advantage of your approach versus a synthetic blood vessel?
Well, certainly, as you point out, there have been synthetic blood vessels that have been on the market since the 1960s and 70s. These vessels are Dacron and Teflon. So these are all materials that we're used to in terms of, clothing and frying pans. And these materials also function as vascular conduits, blood vessel replacements. But some of the limitations of these materials are that you can't really use them in smaller diameters, like for heart bypass, because they clot, because it's plastic. And if you put a tiny plastic tube in the bloodstream, it does clot. But in addition, because these materials are plastic and they don't degrade, they're also subject to infection. So bacteria can adhere to these plastics, and then it's very hard for the body to clear them. So we believe we have some advantages, with respect to clotting and with respect to infection. But I would also say that our vessel is easily available. So like a plastic graft that a surgeon can pull off the shelf, Our vessels are stored in the refrigerator, and literally the nurse or the physician can pull it off the shelf and open the packaging and use the vessel when they need it.
How long does it actually take to develop one of these vessels to the point where it can go and sit in the refrigerator and be ready? What's the timescale?
Well, our whole manufacturing process is about 10 weeks, 10 or 11 weeks end to end. From the time we take a small vial of cells, just 1 or 2 million cells, and thaw them and grow them out. We then use that small vial of cells to grow batches of vessels, 200 at a time. So we grow 200 blood vessels at a time, and then that growth process takes a couple months. Then we decellularize them, and then we test the batch. And if the batch passes, then we send it out to hospitals and clinics.
How big is a batch? How many are we talking about?
So it's 200 vessels at a time, and each vessel is about 40 centimeters long and 6 millimeters in diameter. So again, what's amazing to me is that we're able to start with a small vial of cells. Each vial is about 1/5 of a teaspoon. It's a little tiny volume. But from those cells, which have tremendous growth capacity, we're able to generate 200 arteries that can help 200 patients.
Now, Humacyte was founded in 2004. You've been at the helm right from the beginning there. This initial spark that you had and the idea that you had coming out of academia, did you overcome any skepticism? Were there people who thought that this might not work, and plus we've had these synthetic ones already for 30 years, why do we need to build something new?
Well, the second part of your question, it's very interesting. The vascular surgery community has talked about the biological arterial replacement, especially for small diameters. They've talked about that as being the holy grail of vascular surgery since before I started working on this. So the clinical need has been clear to surgeons for decades. But yes, was there skepticism about whether this would work? Absolutely. And when I first started writing, when I was still in academia and was writing grants to the National Institutes of Health to try to support this research, there was a lot of skepticism that maybe this wouldn't work, even when we had shown initial proof of concept in animals. So it did take some convincing, that's for sure.
Was there a particular point along this journey, now that you've got some FDA approvals, and was there a point where you just got that boost of confidence and said, okay, I'm sure now that we're on the right track, everyone's going to see that this is going to work?
Well, I guess if I was going to talk about one point, I guess I would talk about after we went into our first clinical trials. So we treated our first patient, who was a dialysis patient, in Poland in 2012. And actually, our first phase 1/2 trials were in dialysis and in peripheral artery disease. And we had, of course, tested the vessels really extensively in the laboratory and in animals, in non-human primates. But when you first go into patients, it really is a whole new world. And what I like to tell people is that I didn't sleep very well for the first year, because you never know how it's going to work in patients until you test it in patients. And that's just the reality. But I guess I would say after a year, when we really saw nothing unexpected and the vessel was just working as we expected, as we had seen it work in large animals, after a year, I thought, yep, we're here. We've arrived.
It was December 2024 when you got your FDA approval for the use of the vessels in vascular trauma. What were the challenges there for you navigating that regulatory pathway, particularly because in that whole period, COVID was taking over, and COVID was what everyone was talking about, and some of these other things maybe got pushed a little bit to the side?
Well, certainly COVID was a challenging time for all of us, including biotechnology companies, because it was hard to get people to come to work. And also, frankly, enrolling our clinical trials really slowed down because a lot of non-emergency care just got pushed off to the side. But I will say that we're very grateful to the Defense Department in the United States the Defense Department, because of our results that we had shown in animals and patients, the Defense Department became very interested in the possibility of having this vessel available for wounded warfighters. And so they actually gave us a priority designation with the FDA in 2018. And that really moved us up the importance scale, I think, for the Food and Drug Administration. And so we were able to work closely with the FDA to design a single-arm trial that would allow us to test this vessel in traumatically injured civilians, which we did in the US and in Israel. There had actually never been any kind of randomized clinical trial, not even a prospective or a controlled clinical trial in vascular injury ever done ever. That's because it's really hard to do. You know, if you have a patient with a major vascular injury from a car accident or a gunshot wound, and they roll into the emergency room, they're literally bleeding to death. And getting consent and trying to do a clinical trial in that setting is extremely challenging. But we had support from the FDA and from the Defense Department, and we moved forward, and our trial showed good results.
It's interesting to hear that about the DoD, because a lot of the people that I talk to, if we're talking about the US government and their impact on the biotechnology industry, we're talking about the FDA, we're talking about the NIH. But it sounds like the DoD could actually be a really important player, at least in some areas of the biotech industry, and getting their help and their support could be important to moving forward.
Well, I certainly think so. I mean, the Defense Department is very mission driven, as we all know and understand, and protecting their warfighters and repairing wounded warfighters is right up on the top of their list of priorities. And, you know, they do have situations in combat that are very difficult to study in typical randomized, prospective, controlled clinical trials, because, you know, war is not like a prospective randomized clinical trial. And so the Defense Department has been very focused on trying to find ways to get products available to treat wounded warfighters in a way that's also consistent with all of the other regulatory frameworks that we have here in the US.
Can the vessels be taken into the field quite easily? Or is there a cold chain that's needed, a hospital that's required?
So our vessels are used, you know, by vascular surgeons and trauma surgeons. So I think that the concept that our vessel would be used literally out in an open cornfield, that's not exactly right. You know, our vessel is used, you know, as part of a surgical procedure. So it's used in a hospital or a forward medical operating location. Our vessels are refrigerated. They're not frozen, but they are stored refrigerated, and they have a shelf life of 18 months. So that means that if there's a refrigerator nearby, which all hospitals and operating rooms have, then our vessel can be stored there.
Talking about combat, and you mentioned before that you had done some work in Ukraine and your vessels had been used for trauma in the Ukraine conflict. Tell me how you came to be involved there. You know, how was it that Humacyte happened to become involved in a conflict over in Europe there?
Well, as I've mentioned, we actually started our clinical trials in dialysis and in peripheral artery disease in Eastern Europe. And the vascular surgical community is actually a pretty small community. There's only, for example, there's only 3,000 vascular surgeons in the entire United States. And the same is true across Europe. You know, vascular surgeons know each other. And so our conduit was well known to many surgeons in areas surrounding Ukraine, particularly Poland. So after Russia invaded in February of 2022, I guess it was, then what happened was about a month later, we started getting emails from surgeons in Ukraine who knew about the vessel and who were seeing so many terrible war injuries and seeing a lot of amputations. And this has been written up in the New York Times, the number of amputations that have been seen in Ukraine. So those surgeons were very interested in whether or not they could get our vessels on a humanitarian basis. So we worked with the US FDA, with their international office, and asked for permission to ship our vessels to Ukraine. We were not approved at this time. So we had to get permission for that. And the FDA responded very quickly within a week. And then we worked out, as you say, the cold chain, shipping the vessels into Kiev. And then the Ukrainian Ministry of Health helped distribute them to other forward operating locations.
And did you have to work with surgeons remotely, or this was handled by people on the ground?
So we did have to work with surgeons remotely. And one of my favorite parts of this story is that, so we do have one of our employees at Humacyte is actually a Ukrainian trained physician who speaks Ukrainian, which is very handy for us, because when we developed training materials for surgeons, we couldn't send any of our staff or any surgeons to Ukraine to train them in person on how to use the vessel. So we had to do all of our training over virtually, over video chat, and we had to do it in Ukrainian. So we developed a set of teaching tools and language-specific materials to teach the surgeons how to take the vessel out of the packaging and the best implantation techniques. And it worked very well because their outcomes in Ukraine were outstanding.
What is the learning curve there? How much training do people have to go through to actually use the vessels that you produce?
Well, I like to say that the training is about one case. If you speak to surgeons about how the vessel handles, again, vascular surgeons and trauma surgeons, they're used to sewing human arteries and veins. They do it all day long. And if you ask a surgeon how our vessel feels, they'll tell you it feels kind of like a normal human artery. So once they get it out of the packaging, which is one of the big training parts of this, but once they get it out of the packaging, it actually handles very easily for them. And I would say that it's about a one case learning curve, and then they understand it and they can work with it very well.
Working with people in Ukraine is, in particular, these trauma surgeons. That's some real world case study that might have taken a long time to collect in the United States. You mentioned you work in Israel as well. How has that data, how has that feedback from the surgeons changed things, or how has it helped you to redefine exactly how these vessels might be used and in what situations they might be used?
Well, I do think, to your point, the outcomes in Israel, where we treated, I can't remember, maybe 4 or 5 patients, and then in Ukraine, where we treated a total of 19 patients, overall, those outcomes were outstanding. And again, for all of those surgeons, we train them remotely. And so I think it shows that, number one, that the vessel can be used fairly easily by anyone with vascular surgical training. But it also shows, to your point, many of these patients sustain terrible injuries. These photos, we can't share a photo on a podcast, but unfortunately, there's some pretty graphic photos of some of the horrific IED blast injuries that some of these folks have suffered in Ukraine and Israel. And being able to restore circulation and save the limb in a very contaminated wound environment can really make the difference between amputation and avoiding that.
This work that you're doing, in particular in a combat zone like in Ukraine, how does that feed back into the mission of the company and the people who are working from Humacyte? They see the real world good that the product is doing. They see the real world good that the asset is making in the world. How does it feed back into the mission?
Well, we've had, I think, the Ukrainian experience, but also even our clinical trial experience here in the U.S. Is very, very inspiring for people. I think seeing that, of the patients that we treated in Ukraine, despite some terrible injuries, there were no amputations. And there were some patients who were not expected to survive. But instead, we have a photo of one particularly badly wounded patient walking out of the hospital. So when people on the ground at Humacyte can see that, that's very exciting and motivating for sure. But I'll tell you, we've also had patients who we've treated in our clinical trials or as compassionate use cases here in the U.S., where I believe we've absolutely been limb and life-saving. And some of these people, for example, we have one woman who we treated in our clinical trials who was a dock worker in New Jersey and was crushed by a crane. Her leg was crushed by a crane. And The injury was horrific, and she sustained terrible wound infections, but the vessel kept right on chugging along, and now she's a beautiful woman working a job and walking around in high heels. So it's when you can see that upfront and personal, it's very important for our employees.
You've got your FDA approval now for the vascular trauma use case. What are your goals as a CEO now that you've got this approval? What's the next step? What do you see moving forward?
Well, we're very excited about our collaboration with Fresenius and about our next indication, which we hope will be in dialysis access. So we just last fall, we reported positive outcomes of a phase 3 study. This was a prospective randomized study where we compared our vessel for dialysis access to the standard of care, which is called a fistula, which is where the surgeon goes into the arm and sews an artery and a vein directly together. So that's the gold standard for dialysis access, but those operations often fail and they don't produce a good blood vessel that patients can use to dialyze. So we showed positive outcomes where our vessel performed better over the first year in dialysis patients overall. So we're very excited about that. We're just as excited about a follow-on study that we're currently enrolling, which is a first of its kind study just in women who have kidney failure. Because interestingly enough, if you do this fistula operation in men, you have actually a much higher success rate than you have in women. So women, maybe because their blood vessels are smaller, have a much harder time getting good, stable dialysis access. And this can be very damaging to their health and their well-being. So we're conducting in women, and we're really focusing on dialysis patients who really struggle with access and who are in and out of the hospital with infections and access failures, because we think we can help these folks.
So we've got trauma, dialysis. You're looking at applications in coronary bypass as well, pediatric heart surgery. I mean, is there anywhere that you're not looking, or an area that you hope to move to that you haven't got to quite yet?
Well, at the risk of sounding like we're trying to do too much, I don't think we are, but we've also been involved in large animal studies looking at using our blood vessel as a delivery vehicle for pancreatic islets. So patients with type 1 diabetes don't make insulin. And the reason for that is that their own immune system kills off the cells in their pancreas called the islets, which make insulin. So their body can't make insulin anymore, and they have to inject themselves multiple times a day. Transplantation of islets has been something that we've been doing for years, but those transplantations have a high failure rate because the islets, after we inject them, let's say underneath the skin, those islets don't get enough oxygen to survive. So we're exploring whether or not we can use our blood vessel basically as a delivery vehicle and put islets on the outside of the blood vessel so that when it's sewn into an artery, those islets get enough oxygen and nutrients to survive. We have some proof of concept studies going on in animals right now that are very exciting. So at some point, it would be great to also look and see if we can help patients with type 1 diabetes.
One of the challenges in bioengineering is always getting up to scale. So, creating the first few vessels, it takes a long time, but over time the manufacturing improves and the scale grows, except the scale of cases for dialysis, for coronary bypass, for vascular disease in general is enormous. How do you scale to address that sort of global need?
Well, you're right. I mean, scaling any biologic therapy, whether it's an antibody or a protein or a tissue, is one of the big challenges across all of biotech. I would say that we've been embracing this challenge for a number of years, and as I mentioned earlier, we do have actually a highly automated system now that allows us to grow 200 blood vessels at a time. And the system is really it's a large incubator. It's about as big as a school bus, And we can run that incubator and run about 5 batches per year. So since each batch is 200 vessels, each of our incubators can grow about 1,000 vessels per year. Right now in our building, we have 8 of these incubators installed. So that means our maximum capacity is about 8,000 per year. However, we've got room just in this building to install another 30 or 35. So we estimate that just in this building, our total production capacity could be 40,000 vessels per year. So from our standpoint, it's really about having the scaled technology, which we've spent years and millions developing, which I believe we have in hand, but also having enough cells, enough starting material to do that. And again, we spent more than a decade developing a very large cell bank. Of multiple donors. So I think we can scale.
This certainly sounds like you're up for the challenge. It sounds like you're up for the scale. Laura, I want to thank you for talking to us today, and thank you for joining us on Beyond Biotech.
Thank you, Dylan. It's been a great pleasure. You have a good day.
Too.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.